So I feel privileged to invite the next speaker, Professor Moshe Oren from the Weizmann Institute. Moshe, many of you are probably familiar with his work on p53, tumor suppressor gene. Moshe is the recipient of the Israel Prize in 2008 for biochemistry, a member of the Israeli Academy of Sciences and Humanities, and a foreign associated member of the US National Academy of Sciences. So thank you for coming, Moshe, and we're looking forward to hear your talk.
Thank you, Ronit, for the kind invitation. And let's see, how do I move the slides forward? I'm a mec ignoramus. Okay. So this is going to be about p53, and a few facts about p53 that will put us on the same page. So, as I think everybody here in the audience hopefully knows, p53 is a potent tumor suppressor, and it's encoded by a gene called TP53. And the TP53 gene is the most frequently mutated gene in human cancer, and there are TP53 mutations in about half of all cases of human cancer. Okay, no, that's not this. And now, okay, how do I get out of this now? Okay, fine. Okay, there we go. So what do these mutations do? So it is well accepted and very rigorously proven that the cancer-associated p53 mutations abolish the tumor suppressor activities of the wild-type p53 protein. And that's the main reason that these mutations are selected in cancer. However, interestingly, about 70% of the p53 mutations that one finds in cancer are missense mutations, which means single nucleotide change, single amino acid change, and retention of a full protein. And that's quite unusual for tumor suppressor genes, which typically the mutations that one finds in cancer result in total elimination of the encoded protein. Now, the majority of the p53 mutations that one finds in cancer occur within the DNA binding domain of p53, and they abolish the activity of p53 as a sequence-specific transcription factor. Now, some mutations are more common than others, and in particular, we have several sites which are frequently mutated and are referred to as hotspot mutations, and these are shown here. And I want to draw your attention to position 175 and 273, which I will be addressing the rest of my talk, and we may mention 248 briefly. These are the three major hotspot sites. Okay, so as we know, p53 mutations abolish the tumor suppressor activity of wild-type p53, but in addition, it has been now extensively documented that some missense mutations in p53, which produce mutated proteins, endow the mutant protein with an oncogenic gain of function. So not only they lose the tumor suppressor activity, but in fact they gain some oncogenic activity and actively support cancer progression. And very often this is associated with accumulation of the mutant protein in the cancer cells. This is an example from head and neck cancer, and you can see abundant accumulation of the mutant p53 protein in the nuclei of the cancer cells, whereas you hardly see any p53 in the normal adjacent cells because they have very low levels of wild-type p53. And typically, accumulation of p53 by immunohistochemistry is a fairly reliable indicator that that tumor has a p53 missense mutation. Okay, but is this the whole story? In other words, are all p53 mutations equal, or are some mutations more equal than others? And what I'm going to tell you is that some mutations are more equal than others, and this is the work, practically entirely the work of a very outstanding young person, Ori Hassin, an MD who is now doing a PhD in the lab. And again, this is just as a reminder, 175 and 273 are the ones that we'll be talking about mostly. And Ori decided to look at colorectal cancer and ask whether there's anything interesting in the pattern of distribution of p53 mutations there. And what he noted is that when one looks at early stages of colorectal cancer, this is the analysis of TCGA data. So in early stages, the 175 mutant is much more abundant than the 273 mutants. And in fact, it's almost like a twofold more abundant. So in early stages, and in general colorectal cancer, 175 mutations are more abundant. But what happens if you now go to advanced stages, stage 3, 4, which is essentially metastatic stages? These are the cancers that colorectal cancer patients die of. And now you see there's an impressive shift in the tendency. And now, in fact, 273 mutations are more abundant than 175 mutations, suggesting either that having a 273 mutation early on predisposes to advanced to a stage 3, 4, to an advanced stage, or alternatively, that a 273 mutation just accelerates the transition from early stage to late stage. This is something that we cannot at this stage distinguish. But anyway, having 273 mutations is worse than having a 175 mutation, and we are talking of those mutations, each of those sites is mutated in about close to 10% of colorectal cancer patients. So we're talking about large numbers of patients. And not only is 273 mutations associated with more advanced disease, but if one looks at those metastatic patients that have those mutations, one can find that patients that have 273 mutation have metastasis not only in the standard sizes such as the liver, but also in distant sizes like the brain, bone, pelvis. And usually those uncommon metastases are also associated with worse patients' outcome. Okay. So not surprisingly, but of course one had to look at this. If one looks at disease-specific survival of colorectal cancer patients, those that have 273 mutations fare much worse than those that have 175 mutations that have a reasonably good outcome. So having 273 mutations is bad. Having a 175 mutation is less bad in colorectal cancer. And okay, but what's the mechanism? Why is there a difference? So to address this question, Ori went to an in vitro system. He took SW480 cells, which is a colorectal cancer cell line, a very commonly used one, in which my colleague Varda Rotter's lab has knocked out by CRISPR-Cas9 the endogenous mutant 53, and Ori overexpressed in those cells either the 273H mutant or the 175H mutant, which are the most common mutations in these sites, and you can see that the expression is comparable. He then did RNA-seq on the overexpressing cells, and what he noted, I don't know if I can use the cursor here, but what he noted in the red rectangle is that there's a group of genes, actually 140 genes, which are selectively upregulated by 273 in those colorectal cancer cells and not by 175. And he defined those 140 genes as the R273 signature. He then went on to ask, okay, is this something that is seen only in those, this particular cell line? So we looked at a number of other cell lines in which he overexpressed 273 versus 175. And as you can see here, this is now another colorectal cancer cell line, RKO, which has wild-type p53. Wild-type p53 was knocked out by CRISPR-Cas9. 175, auto-73 overexpressed. And you can see that 273, but not 175, is upregulating representative genes from the signature. So this is not just a single cell line. It's something that can be demonstrated in multiple cell lines. And this is overexpression. So we asked, and in fact, one of the reviewers asked, can you show it also for endogenous mutant 53? So Ori adopted RNP-mediated CRISPR-Cas9 gene editing to take cell line HCT116 colorectal cell line, which has a wild-type p53, and replace the wild-type p53, knock in either the 273 mutation or the 175 mutation, and this is just showing 5 representative clones from each of the knock-ins. And reassuringly enough, he showed that also in this endogenous mutant p53 background, 273, but not 175, can upregulate representative genes from the 273 signature. Okay, so this is all cell lines. Is it relevant to tumors? Okay, why am I going here? Okay, fine. So, okay, this is all cell lines. So is it also relevant in tumors? So Ori went to the TCGA data, and he compared the R273 signature expression in tumors that have R175 mutations and tumors that have 273 mutation. And what he found is an extremely high correlation between tumors that have a 273 mutation and expression of this signature. So this signature is distinctive of tumors that have this mutation, not only in the cell line, but also actually in real human colorectal cancer. Okay. So instead of now looking at the mutation, Ori looked at what happens to colorectal cancer patients that express this signature. And again, expression of this 273 signature at high levels is associated with much worse overall survival than having a low expression of the signature. Just to clarify, the signature is expressed also in tumors that have other mutations. So it's not that you have a 273 signature. We got the signature by overexpressing 273, but we now know that there are some mutations that are driving the signature and some mutations are not driving the signature. And maybe an important point to make here, what we see here is associations, right? When we see a particular gene signature in human tumors, we don't know if this is just a correlation or whether there is a causative relationship. The in vitro experiments tell us that indeed the 273 mutation is driving the expression of the signature and presumably contributing to this worse patient outcome that we see in real patients. Okay, maybe I'll skip this one because I want to rush further. So Ori characterized the 273 overexpressing cells compared to the 175 and asked how do they differ in vitro, and he found that having a 273 mutation causes a largely increased ability to migrate in a transwell migration assay. It largely enhances the ability to invade the metrogyl layer, again in a transwell invasion assay. So having this mutation in colorectal cancer cells makes them more migratory, more invasive, and also more resistant to a variety of stress conditions, including chemotherapy, which I'm not showing here. So does this in vitro different in migration and invasion have any in vivo consequences? So usually migration and invasion are taken as an indication for increased metastatic capacity. So Ori tested this directly in two ways. One is the quick and dirty way, which is a tail vein injection, experimental metastasis. It's really not metastasis, it's more testing lung colonization. And again, you can see the cells that express the 175 mutant colonize the lungs not very efficiently. This is GFP labeled cells, whereas having a 273 mutation results in an increased ability to colonize the lung and this kind of lung metastasis. So yes, this enhances metastatic capacity, but perhaps the more important experiment is in D here, where Ori injected these cells into the cecal wall, the wall of the cecum, and they form a primary tumor, and then they metastasize, and 7 weeks later he sacrificed the mice. And with 175, there were, even though the numbers were small, they are quite striking. With 175, in this system, there were no metastases at all. This is a more physiological colorectal metastasis system. With 273, we saw metastases in liver and lung and peritoneum. Okay, so getting close to the end. So what does it all tell us? So interestingly, several years ago, a large group, international consortium, defined subtypes of colorectal cancer, molecular subtypes, and this is called the consensus molecular signature, CMS in short, and they defined 4 subtypes, and the one that is the worst is the one that's called CMS4, and this is defined as a mesenchymal colorectal cancer type. It's associated with increased EMT, increased inflammation, and has increased TGF-beta signaling and has the worst patient outcome. And when Ori compared the 273 signature with the signatures that defined each of the 4 molecular subtypes, As you can see in the blue line, there was a very strong correlation between the 273 signature and the defining CMS signature, telling us that the 273 signature that Ori identified in vitro experimentally and is driven, at least in vitro, directly by the 273 mutant, is strongly correlated with the defining CMS4 signature, which defines the worst class of colorectal cancer. Okay, so parenthetically, so what about R175? Also a very common mutation. And in fact, R175 is also associated with a specific subtype, in this case CMS2, which is a less aggressive one, and it's more the classical one, Wnt-driven MYC overexpression, which usually is less aggressive. Okay, so conclusions. P53 mutation sequence matter, and the point that I want to make here, why I'm bringing it up, for most researchers and also oncologists, when they look at P53, the question is it wild type or mutant? And if it's mutant, that's good enough, I mean, or it's bad enough. But that's the end of the story, and I think that's true for most of the scientific community. And you see studies all the time comparing wild type P53 tumors to mutant P53 tumors and showing that it works on this and not on that. And in fact, very often there's a lot of noise in the mutant group, but you get enough statistical significance. What Ori is trying to tell us is that not all mutations are equal, and in fact, knowing what a particular mutation is associated with could be very important for figuring out how to deal with that particular cancer patient. And one mutation is not the same as another one. And okay, so I think I skipped the 248, but 248, just bottom line is 248 is very similar in its behavior to 273. Okay, maybe I'll just show this. And if we look at the effect on survival, red is 175, better outcome. 273 and 248 actually are almost indistinguishable. So we have two major hotspot mutations which are enriching the 273 signature. We defined it with 273, but 248 is also driving it. And this signature is associated with worse patient outcome and with a different type of colorectal cancer involving different signaling alterations. So in fact, knowing which particular mutation is present in the cancer patient, okay, so maybe I'll just read it. So different P53 hotspot mutations have a different impact in colorectal cancer. Different P53 hotspot mutations orchestrate different transcription programs with different impact on the biology of the tumor and the prognosis. And we propose that different P53 mutations might be selectively advantageous in different stages of colorectal cancer progression. In fact, I didn't show you, but it looks like 175 has an advantage early on because it's associated with activation of the enhancement of beta-catenin signaling, which is the initiating event in colorectal cancer. 273 and 248 mutations drive tumor progression, and P53 mutation status may therefore dictate the extent to which a particular CRC tumor will be responsive to a particular molecular targeted therapy, because the signaling landscape of tumors that have 273 mutations and 248 mutations is very different than those that have 175 mutations. Okay, so this is a picture of the group, and I want to thank again our contributors. So as I said, almost all this work was done single-handedly by Ori, but he got a lot of support and advice by many other people in the lab and by our collaborators, which are all listed here, and thank you.
Thank you, Moshe.
Cyril has a question.
Super interesting.
It's a great idea. We have not. The closest I can say, which is really not addressing your point, but with 175, the seems to have a different effect on the immune system than 273. And in fact, the tumors that have 175 are in fact more immune cold. So we think that, and in fact the 272 tumors are inflamed, but they're inflamed in a way that promotes cancer progression rather than cancer inhibition. Just from this analysis of TCGA data, and also got a database from Memorial and Sloan Kettering. So, but we cannot trace the same mutation in the patient over years. So all I can say is what I said here. It looks like 175 is probably advantageous. 175, okay, maybe I can say one more thing, which we looked at the variant allele frequency in those tumors. And in fact, the tumors that have the 175 mutation have a higher variant allele frequency. In other words, they are more tend to be homozygous, suggesting that in fact maybe the 175 is mostly a loss of function mutation in this setting, whereas the 273 is a gain of function that can exert its effect even if wild-type p53 is still retained. But that's all I can say. And again, it looks like 273 is accelerating tumor progression based on these correlations. We haven't done it in a mouse model because that would be the way to really stepwise do it correctly and answer your question the way it should be answered. Yeah, okay, yes. The answer is yes, okay, but yes to what? Okay, so some mutations are definitely oncogenic drivers. The question of, the term driver is a little bit, I think, not, well, controversial, maybe confusing, because when you think of a driver, it's something which is actively driving, like RAS, right? RAS EGF receptor. We think that some of the p53 mutations are, I would call them enablers. I mean, they are not driving the cancer, but because they are there, they enable real driver mutations to drive the cancer forward. And I think a classical example in vitro is if you overexpress RAS, p53 is activated and they drive the cells to senescence. If you get rid of p53 or you have a mutation that gets rid of p53 functionality, then RAS can drive this cancer. Very simplistic, but then, I mean, the difference between drivers and enablers is kind of fuzzy, but in both cases, one would argue that they contribute to the cancer, right? And there are passenger mutations, definitely. There are many passenger mutations. In any gene, you find passenger mutations. p53 has been sequenced ad nauseum, and people have found pretty clearly any residue can be mutated. Usually the ones that are found in very few tumors, if one tests them functionally, they retain wild-type p53 activity. And for those who, I don't know how many people know the SW480 cell line intimately, but in fact the cell line has a 273 mutation and the 309 mutation, which is noted, but nobody cared about it. And one of the reviewers asked us, maybe what you see is because of 309. So we expressed 309. It works just like wild type. It kills the cells and you use the apoptosis. So in fact, it's a passenger mutation. It's there, but it has nothing to do with the cancer. לבריאות.
More of a futuristic question. Here has been oncology, prognosis, and therapeutics. Can you comment on it? Maybe the mutation status is the point where we should put our effort. Also, maybe pathology. According to the mutation.
Okay. Okay, so this is it's a big question, right? Therapeutic targeting of p53 is the whole world, and people are doing that, and that takes us in a different area, including the delivery of p53, wild-type p53 by nanoparticles in tumors, which has been shown in mice to work. I don't think that's perhaps, but that there might be better ways and easier ways, but That's doable as well. I'm just thinking of what Dan said, right? You can target wild type p53 into specific cancer cells, and at least in mice, it has an effect. But relating to the maybe the more specific question, I would suggest that immunohistochemistry is necessary but not sufficient. Usually when you have a missense mutation that has a gain of function, it also is accumulated. That's more or less a paradigm in the field. So if you see stabilized mutant p53, you would want to sequence it. And often this happens because all the gene panels there, let's say, right, Oncotest, Foundation Medicine, all those standard gene panels have p53 in them. So they come back with p53 mutations 50% of the time. The problem is that then the oncologists say this is unactionable, very sad, let's look at what else is there. And what we are trying to move is to say, let's look at the p53 mutation and it will tell us something. It's not something that, okay, non-actionable, forget about it.
Thank you.